Hello, my name is Mia Albinson, and my research project is developing a machine learning model to detect the chemotaxis of C. elegans for an at-home cancer risk assessment. As I'm sure you know, cancer is a genetic disease caused by the rapid cell growth of mutated cells, and early detection is vital to combat the ever-growing nature of this disease. Tumors are more likely to be localized during initial stages, and this localization significantly reduces the risk of metastasis, thereby increasing the effectiveness of treatment and significantly improving survival rates. Additionally, localized tumors often allow for less invasive and less intensive treatment strategies, which can enhance a patient's quality of life during therapy and potentially shorten the overall treatment duration. These approaches may also reduce the risk of secondary health complications commonly associated with more aggressive treatments such as radiation and chemotherapy. One model organism that is actually used for cancer screening is C. elegans because of their surplus of G-coupled proteins that give them very advanced olfactory systems. This allows them to detect the volatile compounds of malignancies found in urine, and their neural networks actually make them attracted to the smells of cancerous urine and inversely repelled by those of healthy urine. Therefore, by studying the chemotaxis, which would be characterized as movement towards or away from a urine sample, one is able to determine if a patient is at high risk or low risk of cancer. One test that actually utilizes C. elegans for cancer screening is the NYSSEF test based in Japan at Harvard Labs, where they found the test to have an accuracy rate of 71.2% after testing on more than 1,600 cancer patients. It was able to detect more than 20 different types of cancer ranging from stages 0 to 1, and is extremely impactful in the advancement screening test, being completely non-invasive procedure. This test only counts for cancer risk type or stage of cancer, and it cannot determine the specific type or stage of cancer, but it is a suggestion to go in for further screenings delays that happen within the diagnosis process. The EndNotes test, the patient sends in their urine sample to a lab, which can increase the time within diagnosis, and can also increase the cost, which is standing now at around that's what I want to improve with my research. This is what I want to improve with my research model. By developing a machine learning model, the patient will be able to increase the efficiency results at home, which would also be able to decrease the cost, making it more accessible. One test that actually utilizes this brings me to my research question: how can machine learning be implemented into the EndNotes test to increase efficiency? My hypothesis is that a trained model will be able to accurately detect the chemotaxis of C. elegans through an inputted image. I tested a model on Teachable Machine using Fox Snow to mimic the presence of C. elegans because of their similar size, and then I painted one half of the petri dish white and collected my dataset. So more snow on the white was trained as positive, while more snow on the black side was trained as negative, and after it was trained, I ran multiple images through and found it to be 100% effective in identifying the side with more snow, and therefore I implemented Teachable Machine through my project. I based the setup of my test off the EndNotes test because I want to achieve similar accuracy. So the X's mark where the urine sample would be placed, and the dots mark where controlled water would be placed. I had roughly 50 microliters of C. elegans either directly onto the X's or dots to simulate a positive or negative chemotaxis. So if there were more worms on the X's, it was positive and moving towards the urine sample, and if there was more on the dots, it was negative. I created 8 positive and 8 negative tests for my first model, and I took a burst of pictures for each test under 2 different lighting conditions to try and highlight the presence of C. elegans. I ended up with a very large data set of roughly 2,000 photos for each category, and I trained 2 other models, one without taking the burst, so I had a total of 16 images per data set, and I did this test to see if a larger data set would affect accuracy, and I created another test with just diagrams. I did this because I wanted to know if the diagram, which has only one variable changing, would be more accurate so that I know that the model has issues in detecting the dependent variable. I inputted each picture into the 3 different models and found the percentage detected correctly for the positive and negative simulated tests. And as you can see in these graphs, it was significantly more accurate in detecting positive tests, while very insignificant in detecting the negative tests, suggesting that it was overdiagnosing. And with the variations between the different dataset groups, it suggests that the model and the iPhone is not able to capture where the C. elegans are and differentiate where they are on the petri dish. My first method for improvement was staining the agar with food coloring. I added 50 microliters of each color into the petri dish. I used 2 different colors because previous research had pointed out to a color preference within C. elegans. So I let the dye sit for 48 hours, and after the time elapsed, I rinsed off the dish with PBS and viewed the worms under a microscope. Upon viewing, it was clear that the C. elegans had undergone slight staining, but it was very insignificant and not visible without a microscope, so therefore negligible in my research. Next, I tried Kamasi protein staining solution and methylene blue to stain the C. elegans. 50 microliters of each dye was added to 200 microliters of C. elegans suspended in PBS. The dye's methylene blue was used because of its ability to stain yolk particles in C. elegans, and Kamasi protein staining was used because of its success in identifying lipid droplet proteins in past research. Once exposed, they were set to sit for 24 hours in the dark, and after they were placed into a centrifuge and to pellet, and then were viewed under a microscope. Although close examination showed signs of coloration, as you can see in the images, the change in visibility is very slight, and in both solutions there is a precipitate formed, the majority being in methylene blue, which would have caused issues in imaging. Finally, Lugol's iodine was used based off its use in previous research to stain and identify glycogen content. So worms were grown on agar seeded with glucose to increase their glycogen content, and then 100 microliters of C. elegans were collected and mixed with 400 microliters of Lugol's iodine solution. And then after centrifuging and pelleting and viewing under a microscope, the worms were seen to be heavily stained, as you can see in the image. However, all of them had died. So this occurred because of the excess iodine caused oxidative stress within the nematodes. So a smaller amount of iodine was tested to see if staining is possible without causing oxidative stress. So this time 400 microliters of C. elegans were mixed with 200 microliters of Lugol's iodine at 2%. And immediately after they were rinsed. So then there was very minimal staining. However, all of the worms had still died, so therefore Lugol's iodine was concluded to be unsuccessful. Overall, my hypothesis was refuted. The machine learning model could not accurately determine the chemotaxis of C. elegans. Although my hypothesis was not supported, this research contributes to the growing field of AI-based biomedical diagnosis by highlighting the challenges of applying machine learning to behavioral analysis. This work highlights the limitations of low-cost, at-home cancer risk screening tools and introduces the risk of overdiagnosis, with my data being profoundly focused on positive detection. Finally, my research suggests that future work should focus on improving the visibility of C. elegans by increasing glycogen content through enhanced glucose in the culture media. Additionally, synchronizing the worms so that they are all in the adult stage rather than in mixed life stages could improve consistency and visibility during image capture. To improve detection accuracy, my findings indicate for manual annotation of the images to use more advanced machine learning platforms. This is likely due to the current model's difficulty in reliably identifying C. elegans within complex or low contrast visual environments. And this is my works cited. Thank you very much for your time.